complementation test is a cis-trans test that is used to determine if a gene is on two different loci or the same loci. If two genes are complementing each other, that means they are on two different loci. This test shows us if two mutations are complementing each other. This is a brief example of complementation, showing two individuals who are homozygous recessive for a mutation at different alleles, and their projected offspring showing a different phenotype due to being heterozygous at these alleles. Just a little background on yeast. It reproduces asexually and sexually. For this experiment, we need to focus on the sexual reproduction to actually show the complementation between the different yeasts. Currently being shown is a normal adenine pathway without any mutations, resulting in white yeast colonies. This pathway shows a mutation on ADE1, which results in red or pink yeast colony pigmentation. This is due to the buildup of AIR. This is a mutation on AED2, which causes the pathway to stop a little bit earlier than the mutation on AED1, but this also results in red to pink pigment due to the buildup of AIR. The final mutation is a double mutation at ADE1 and 2. Like the two mutations before, this also results in a red pigmentation of the yeast. For my hypothesis, I predicted that 2 of the 9 yeast mixes would produce only white yeast, resulting in complementation, and the rest would be variations of red and pink due to mutations before adenine could be produced. For this experiment, we used 5 different strains of yeast, 3 of which had a mutation and 2 of which were unknown, along with the other supplies listed above. For the complementation test, we mixed each strain of yeast with one another and viewed the results. To start off the experiment, I labeled my agar plate as shown above, to prevent confusion and keep each yeast mix in order, and to make the observation of each yeast easier. The next step is to mix our yeast. I obtained each strain of yeast, and for each one, I used a sterile loop to scoop out a colony of yeast from the culture and placed it into the correctly labeled spot on my labeled agar plate. After all the mixes are placed onto the labeled agar plate, they are given to a professor to be incubated at 24 degrees Celsius for 24 to 48 hours. Right away, you can see that 7 of the 9 mixes are red, showing that there was a mutation in the yeast, but only 2 of the 9 mixes are white, showing complementation. There are also 4 spots of mold growth, primarily around all of our white yeast mixes. You can also see in some of the mixes where both sexual and asexual reproduction took place, causing one corner of the yeast to be all white and the other side all red, with a pink in the middle. Shown here is a better representation of where mutated strands are and where complementation took place. Here you can see any mix containing mutated yeast are B, C, and E strands, all circled in blue, while the unmutated strands were A and F, all circled in pink. The results I got from my yeast complementation test matched my prediction that only 2 mixes would show complementation, since when mixing A and F, both yeast cultures were white. Now some of my mixes are definitely open for interpretation, due to the fact that the agar plate grew mold may be affecting the reproduction of yeast, and some of the yeast wasn't mixed well enough to show a good amount of sexual reproduction. For a future try, I would make sure that all the yeast was mixed completely for more accurate results.